I'm Olga. I'm the commercial specialist at the TNAC, so the Native Antigen Company, which is part of LGC Diagnostics and Genomics. And in this next few slides, I wanted to introduce you to LGC, especially LGC Diagnostics and Genomics, and show you our broader offer. I would also like to introduce you to the Native Antigen Company and present some interesting facts, including how we contributed to stopping the recent pandemic. All right, let's start. LGC Group consists of four main sectors. You might recognize LGC standards. They are pretty well known, but there's also Assure, Axolabs, and what we're going to talk about today is diagnostics and genomics. Within LGC Diagnostics and Genomics, as the name suggests, we have everything that you may need to design molecular or serological assay, from NAC chemistry to quality measurement tools. But what we're going to focus on today is the antigens and antibody section. As you can see at the product as well, all these can aid to your vaccine development research, especially our protein serology section that we're going to focus on right now, so reagent components, which is basically part of the Native Antigen portfolio. Right, so the Native Antigen Company. We are mainly known as the global player in the vaccine development and IVD world. We specialize in producing the high quality proteins, mostly focusing on infectious diseases. At the moment in our catalog, we have over 1,400 antigens, although I believe this number is actually closer to 1,500 at the moment, because we are adding new antigens every week to our portfolio. And if you cannot find what you're looking for, don't worry, just contact me and we can produce pretty much anything that you would like at a certain moment as a part of our custom manufacturing process as well. We offer 3 expression systems. The most used is our mammalian HEC293 cells, so our mammalian expression system Vir2E. We also have insects and E. coli expression systems. We are also ISO 9001 certified and located in Oxford, UK. Our spacious UK facility allows us for rapid scale-up of any protein that you may need quickly, and then completely saturating the market. As you can see on the picture here, we use bioreactors. What you can see being manufactured at the moment, I believe, could be our virus-like particles that will be developed a bit more by my colleague in a second, so you will learn about them a lot more. But we also produce on a gram scale rheumatoid factor, and can pretty much scale up anything that we have in our portfolio. Right, about saturating the market. So you might recognize the Native Antigen as the first company who ever introduced SARS-CoV-2 S1 and S2 antigen on the market. We were first in February 2020, and it was produced in our most used expression system, HEC293 cells. And this is the fun fact that I wanted to tell you, our contribution to taming the spread of COVID-19. Well, we saturated the market completely with these antigens. They were in many diagnostic assays. We all remember the lateral flows that we had to take before going to shop or work or hospital. Well, they were also very needed by vaccine developers around the world, especially by our neighbors, Oxford University. And they used it to support their vaccine trials at the Jenner Institute. And we like to think that through that, we contributed to stopping the pandemic a little bit. Thank you very much. That's the end of my presentation.
Let's have a closer look at some selected products from the Native Antigen Company now, and how they can advance vaccine development. My name is Holger Schuhmann, and I am the head of department of the downstream processing team within our research and development group here in Oxford. I have a Ph.D. in biochemistry, and after postdocs in Germany and Australia, I joined the company in 2013. So I've been with the company for almost 12 years now. Now my team develops the purification protocols and formulations for almost all our products. In the second part of our presentation, I would like to introduce to you our virus-like particles, or short VLPs, and present our dengue and chikungunya VLPs as examples. After that, we will have a closer look at our trimeric influenza hemagglutinin. Let's start with the VLPs. According to this definition, borrowed from Wikipedia, virus-like particles can be defined as particles consisting of the structural proteins of viruses, but do not contain other proteins such as non-structural proteins or genetic information necessary for replication. They look similar to the original virus, but contrary to the virus, they cannot replicate, and therefore they cannot sustain an infection of an organism. This makes them an interesting choice when working with a live virus is not possible, for example, because it is too dangerous. Most VLPs assemble spontaneously when the individual structural components are expressed in a native or native-like environment, for example, in a mammalian cell. Others can even assemble when their individual components are mixed in vitro. And lastly, VLPs can be obtained from many different viral families, including pathogens to a huge variety of species. So, which VLPs are of interest to us for vaccine and diagnostics development? There are different types of VLPs that can be used for different purposes. Unmodified VLPs can be used as antigens or immunogens, whereas transpackaged VLPs as a replacement for viruses in high-risk PRNT assays. PRNT stands for plaque reduction neutralization test. For some viruses, it is the gold standard for diagnostics, but unfortunately, it often requires a culture of a virus, which can be dangerous. Pseudotype VLPs can also be used as a virus replacement in PRNT assays, and additionally for transmembrane protein enrichment. And lastly, carrier VLPs are used as scaffolds for chemical and genetic fusions of immunogens and for drug delivery. To keep it simple and for the sake of time, we will focus on unmodified VLPs in this presentation. So, why are unmodified VLPs preferred as vaccines and in serological assays? Well, our immune system has evolved to recognize repeating molecular patterns associated with pathogens, and also to respond to immunogens of a certain physical size, such as viruses. For example, an IgG molecule contains 2 binding sites for the same epitope, whereas an IgM molecule even contains 10. As a consequence, many neutralizing antibodies, meaning antibodies that prevent infection, attack homo-oligomer junctions present on the surface of viruses. Unmodified VLPs possess these homo-oligomers and their associated repeated molecular patterns, whereas single protein units do not. In addition, VLPs are of a size that is required by the immune system to react, another property that sets them apart from single protein units. Therefore, unmodified VLPs are superior to protein monomers. I would now like to introduce to you some of the products the Native Antigen Company developed. They are just a tiny snapshot of our catalog, but I think they highlight some of our capabilities quite well. The first group of products are our dengue VLPs. Just as a recap, dengue virus is a member of the Flaviviridae, with 6.5 million reported infections per year, although a total of up to 390 million infections per year is estimated by some models, since most infections go unreported. It is a single-stranded, positive-sense RNA virus, and it is enveloped, which means it is surrounded by a bilayer lipid membrane. It comes as 4 different serotypes, dengue 1 to 4. The envelope anchors 2 glycoproteins, pre-M and E. An expression of these 2 proteins is sufficient to drive VLP formation. The E protein forms trimers of dimers, and these form rafts that cover the viral surface. We built the expression construct by simply taking the part of the dengue virus genome that encodes the pre-M and E protein and drives secretion by the Japanese encephalitis virus signal sequence. As Sang and co-workers had reported earlier, expressing the full-length dengue E protein would fail to yield secreted, soluble VLPs. Therefore, the last 20% of the E protein, which is roughly the transmembrane and intraparticle portion, had to be replaced by a corresponding sequence of the Japanese encephalitis virus, a closely related other flavivirus. Using this approach, we were successful in making VLPs from all 4 serotypes, dengue 1, 2, 3, and 4. For many years, we made them using transient transfection of HEC293 cells, but recently we created stable cell lines for the production of those VLPs. This enables us to greatly increase the possible output, especially with our new bioreactors, such as Olga has shown earlier. On a molecular level, there is no difference between the previous VLPs from transient transfection of cell culture and the new version from stable cell lines. We are able to analyze all our VLPs using field flow fractionation, combined with the UV detector, differential refractometer, and a multi-angle light scattering detector, short MALS. Here I shows one example, a dengue 2 VLP batch produced by transient transfection. In the green line here, the UV, we can see one main peak around 43 minutes, followed by a smaller shoulder at 45 minutes. According to the data from 2 runs, the first peak consists of monomeric VLPs, as the observed molecular weight of the first peak agrees well with the expected weight of 5 to 6 million Daltons, and the observed hydrodynamic radius also agrees with the expected one of around 20 nanometers. The second peak seems to consist of higher order oligomers. The molecular weight roughly corresponds to trimers. I'm not sure if these are aggregates, as the molecular weight is a bit too small for it, and the peak or the shoulder is a bit too sharp. The majority of VLPs are single particles though. In any case, it shows that we are able to distinguish between VLPs and higher order oligomers and aggregates. I have to confess that I had to look hard for a dengue VLP sample that demonstrates our analytical capabilities, using FFF and MALS, as most dengue VLP samples look like this. Pretty much 90% is VLPs, no observable aggregates, and only very few fragments. This is from a dengue 2 VLP batch produced by a stable cell line, and it basically only shows one nice peak, with the signals from the UV detector, the differential refractometer, and the light scattering overlaying nicely. Okay, so we have established now that our VLPs are particles indeed. But how do they actually look like? Pretty much like this. In cooperation with Oxford University, we were able to obtain electron microscopy pictures from all dengue VLPs we provide. As we can see on this slide, for all 4 serotypes, we have really nice particles, resembling the dengue virus. This means our VLPs look nice, but do they also perform well? In order to address this question, Stefan Metz and co-workers compared our virus-like particles to live dengue virus with regards to protein content, epitope availability, and how patient serum reacts with them. As a first step, they simply analyzed the VLPs and viruses by SDS page, followed by either Comasi stain, on the left, or Western blot using an anti-E or anti-pre-M antibody, respectively. As we can see in the A panel, the protein sizes match very well between the virus and the VLPs. Looking at the Western blot, there are slight differences in the E to pre-M ratio, even though some of those might be explained by different amounts of protein loaded onto the gel. They then went on to compare the viruses and VLPs by ELISA, analyzing them against a large panel of human and mouse-derived monoclonal antibodies. Those antibodies indicated in gray are serotype-specific, whereas those in black are cross-reactive for several serotypes. Some of these monoclonal antibodies are known to recognize quaternary epitopes. That means epitopes that span amino acids, which in the sequence are not adjacent to each other, but are only close to each other when the protein is folded correctly. And in the case of a protein complex, the complex is assembled correctly. Those antibodies against quaternary epitopes are indicated by a small q in the graphic, although I am not sure if it's visible on the screen. My apologies for this. The experimental setup means that it is a qualitative analysis rather than a quantitative one, so we can't directly compare the absorption values for a given VLP and a virus. But we can compare the pattern of which antibodies bind and which don't. And that reveals a very good agreement between the virus and VLPs for any given serotype. Compare here, here, these, and these. Antibodies which bind strongly to the virus do so as well in the VLPs. And those that don't bind, they don't do either, neither in the virus nor in the VLPs. All this indicates that the epitope presentation in the virus and the corresponding VLPs is essentially the same. Finally, Metz and co-workers tested how efficiently convalescent patient sera can be depleted of dengue-specific neutralizing antibodies. When serum containing neutralizing antibodies is mixed with virus, the binding of those antibodies to the virus basically prevents it from infecting a cell. However, if the serum has been depleted of neutralizing antibodies, for example by a prior incubation step with immobilized virus or VLPs, which is what they've done here, the serum doesn't prevent the virus from infecting cells anymore. In the upper panel, they first determined the dengue virus-specific IgG levels of depleted and undepleted sera by ELISA, showing that VLPs and viruses, the red and green lines respectively, perform exactly the same in depleting the patient serum of dengue-specific IgGs, as opposed to example for bovine seroalbumin in purple. They then performed neutralization assays, where the depleted and undepleted sera were tested for their ability to prevent dengue infection of a cell culture, by first mixing the different sera with the viruses, and then testing these viruses for their ability to infect cells in cell culture. Whereas undepleted and BSA-depleted serum is effective in preventing infection, because these still contain all the neutralizing antibodies, the serum is unable to prevent infection when it was depleted by either the virus itself or the corresponding VLPs, since both would have scavenged all neutralizing antibodies. Taken together, all this data that I presented so far shows that our dengue VLPs are a great substitute for dengue virus in all applications where using the live virus is impossible, inconvenient, or too dangerous. Our second case study, which will be a little bit shorter, are our chikungunya virus-like particles. Chikungunya is a member of the Toga viridae family. 500,000 cases were reported in 2023, but models predict that the total number of new infections is up to 35 million per year, again, as most infections go unreported. It is an enveloped, positive-sense, single-stranded RNA virus, with the structural proteins being the capsid protein, and the envelope embedded glycoproteins E1 and E2. E1 and E2 form heterodimers, which are then trimerize to form trimers of dimers. We constructed the expression vector by basically taking the region of the chikungunya genome that encodes for the structural proteins and cloned it into the expression plasmid. When HEC293 cells are transiently transformed with this construct, chikungunya VLPs are produced and exported into the cell culture medium. The Kumasi-stained SES page shows the two glycosylated E proteins running closely together, basically running as a single band, and the capsid protein around 30 to 32 kilodalton. To check if what we purified indeed consists of virus-like particles, we cooperated with Puxano, a company specializing in structure-based protein design and structural biology services based in Ghent, in Belgium. Together, we obtained high-resolution cryo-electron microscopy pictures of our chikungunya VLPs, and the scientists at Puxano were able to do single-particle analysis of them. This yielded high-resolution structural features maps, which I would like to show to you now. First up are the full and the cross-section image of the electron density map of our VLPs. The trimers of the E1, E2 heterodimers are very nicely visible on the surface. In the cross-section image, you can see these trimers of dimers anchored on the outside of the lipid bilayer membrane, the structure here, and a layer of the capsid protein just underneath the envelope, as you can see here. Based on this single particle map, the scientists at Buxano were then able to isolate the structure of individual protein complexes and model secondary structures such as transmembrane helices crossing the bilayer. The fact that they were able to obtain these high-resolution images nicely illustrates the exceptional quality of our chikungunya VLPs. For our last example, we will leave the virus-like particles and focus on another group of proteins which the Native Antigen Company successfully managed to stabilize in their native conformation, the influenza hemagglutinin, or short HAs. These proteins are present as trimers on the surface of the influenza virus, immediate adhesion to the target cell, as well as cell entry, and are therefore indispensable for infection. This also makes them an interesting target for diagnostics and vaccine development. They contain two main regions, HA1 and HA2, and trimerization is mediated by the transmembrane anchor. However, when this transmembrane anchor is included in the construct for the expression of recombinant proteins, it leads to insoluble product because the HAs aggregate. We therefore substituted the transmembrane anchor with a T4 fold-on domain, a soluble trimerization domain from the bacteriophage T4. During development of these products, we used native page, which separates proteins by size, but doesn't break up complexes as denaturing SDS page does, to analyze different constructs with and without the T4 fold-on domain. We can clearly see that proteins without the T4 fold-on domain, such as these ones over here, are present as monomers and trimers in varying ratios. However, those constructs that include the T4 fold-on domain, such as this one and these three, consist of more than 95% trimers, as you can see in these lines. Over the years, we were able to add many influenza strains, A and B, to our catalog. I won't go through this list in detail. I just want to show to you that we are able to provide a huge variety of HAs, and we strive to update this list every time the influenza vaccine composition gets updated for the northern and the southern hemisphere. In addition to the HAs, we also provide a similar number of influenza virus neuraminases, NAs. Again, as soluble proteins in their native conformation, which is a tetramer. We are able to provide influenza nuclear proteins as well. If you require a HA, an NA, or a nuclear protein from a strain that is not in our catalog, please do contact us for a custom quote. We have lots of experience with influenza proteins, can also customize your proteins with regards to tags, for example. We've done a lot of custom development and production over the last couple of years. But are our HAs any good? Well, let's start answering this question by looking at the oligomeric state again, this time not by native page or FFF models, but by size exclusion chromatography multi-angle light scattering analysis, or short, SEC-MALS, as we also have a HPLC unit in addition to our FFF. Based on SDS page, a HA monomer would have a molecular weight of around 100 kilodalton, depending on the exact strain. As we can see in the chromatograms, the HAs elude in one single main peak, sometimes accompanied by a smaller trailing peak, which might indicate a different glycosylation pattern, and by a huge last peak observed towards the end of the run. This simply indicates that the run is finished. Looking at the table with a molecular weight measured by the light scattering, we see that the main peak corresponds well with an expected size for the trimer of 300 kilodalton. And it also shows that this trimer constitutes the vast majority of the protein present. The smaller trailing peak, with a molecular weight reminiscent of a dimer, might either indicate an intermediate in the trimer assembly or a trimer of differently glycosylated proteins, as the LCS pages for these proteins don't show any contaminants. We also tested a range of human neutralizing monoclonal antibodies with known specificity against a panel of our trimeric hemagglutinin, which we produced. This set was part of a custom project, which is why I am not able to reveal the exact identity of the strains used here, unfortunately. The antibodies had specificity for H1 type hemagglutinins, H3 type HAs, or for HAs from a wide range of strains. In this first slide, only H1-derived HAs were tested. And as we can see, they were well recognized by the broad-range clones, but not by the H3-specific clones. The results for the H1-specific clones reveal that they might not be able to neutralize every HA, but display a narrower strain specificity. We see something similar when a set of H3-derived hemagglutinins was tested. They were all well recognized by at least one of the broad-range antibodies, although one of them only recognized two of them. Please keep in mind that the antigens were presented in two different concentrations, so each HA is represented by two bars. None of the H1-specific clones recognized our H3-derived HAs, and recognition by the H3-specific antibodies was quite strain-specific again, similar to what we've seen in the previous slide for the H1-derived proteins. Taken together, these results show that our HAs are very well suited for the characterization of anti-influenza antibodies. So I hope I was able to convince you that the Native Antigen Company is a provider of high-quality antigens for vaccine development. We specialize in providing proteins and complexes with native-like structures, and I showed you 3 examples for this. Our dengue virus-like particles from all 4 serotypes, which are intact particles indeed and behave like the virus in neutralization assays. Our chikungunya VLPs, which assemble into structures basically identical to the virus. And finally, our influenza virus hemagglutinins, which are soluble proteins and are stabilized as trimers. Those were proven to interact with a range of neutralizing antibodies. Thank you very much for your attention.